Welcome to our latest news channel. I'm your reporter Shannen today. After the COVID-19 pandemic, the biomedical field is more introduced to the public and more people know about what is biomedical science. Hence, the topic we are discussing today is microbial sensors, which is widely used throughout the process in COVID-19 pandemic. Without further ado, let's welcome our journalist Nick to share us with a few local scientists to know more about the biosensors.
Thank you, Shannon. Hi everyone, I'm Nick. Today I will be interviewing a few scientists to share their knowledge on microbial sensors. So, Dr. Lee, can you tell us more about biosensors?
Let's start with a simple one. What are microbial sensors? They are devices designed to detect substances through specific biological reaction, utilizing elements such as the enzyme receptor, antibody, or immunobiosensor. These sensors have a wide range of applications in various fields, including environmental monitoring, medical diagnostic, food safety, and industrial processes.
Such a clear explanation. Thank you, Dr. Lee. Could you give us the types of microbial sensors and perhaps some examples, Dr. Lauren?
Sure, but before that, let me explain the essential properties of biosensors.
Firstly, a biosensor needs to be specific to the analyte in which it is interacting with. Besides that, it needs to be durable to withstand repeated usage for a long period of time. It should also have an independent nature in which environmental factors like temperature and pH will not influence its function. It needs to be stable to ensure that the results obtained align to the concentration of the analyte being used. Lastly, it should also be of a reasonable size to ensure that it can be used remotely. Dr. Jia Xuan, would you like to introduce the types of microbial sensors?
Okay. Microbial sensors can be categorized by their detection mechanism and the microorganism they use. Here are some type and example of each microbial sensors that are commonly used. First, electrochemical biosensors will measure the changes in electrical property due to microbial activity by using electrode, and it is based on the reaction of enzymatic catalysts that generate electrons. Such types of enzymes are named redox enzymes. Enzyme of electrical sensors include conductor matrix sensors, which are used for detecting heavy metals in water using E. coli, and parametric sensors use E. coli to measure glucose level. Potential sensors detect toxic substances, and water-metric sensors for detection of organic pollutants like phenol. Moving on, optical biosensors is a device that uses light-based methods for detection and uses an optical measurement principle. The light absorbed or emitted as the result of a biological process is used in optical biosensors. Examples of optical biosensors include fluorescence-based sensors that uses genetically modified organisms that emit fluorescence in the presence of specific analytes, such as heavy metals, and another example of it is bioluminescent sensors that use bioluminescent bacteria that emit light in the presence of specific chemicals to detect toxic pollutants, useful for monitoring water quality. Other than that, whole cell biosensors help to utilize entire microorganisms to sense and detect environmental changes. Piezoelectric biosensors detect changes in mass on a piezoelectric crystal caused by microbial activity, useful for detecting pathogens. Next, thermal biosensors measure the heat release and any heat changes during the biochemical reaction, and it can be used to monitor fermentation system, wastewater treatment, and food technology. To develop a microbial sensor, it involves several key steps. First step is the selection of microorganisms, where we have to choose microorganisms that are highly sensitive to the target analyte so that it's effective. Then genetic engineering is involved when microbes are modified to produce detectable signals such as fluorescent or electrical changes in response to the target substance. After that, we combine with electrochemical or optical detection methods to create a complete sensor system where integration with detection technologies is involved. Last key step will be optimization. This is to improve sensitivity, specificity, and response time through further engineering and testing.
After all the introduction of microbial sensors, here comes the briefing on how microbial sensor works. A biosensor mainly consists of two parts, the biological part and the transducer part. The biological part constitutes of enzyme antibodies which interacts with the analyte particles and induce a physical change in these particles. The transducer part collects information from the biological part, converts, amplifies, and displays them. In order to form a biosensor, the biological particles are immobilized on the transducer surface, which act as a point of contact between the transducer and analyte. When the biosensor is used to analyze a sample, the biological part specific to the analyte molecule interacts specifically and efficiently. This produces a physiochemical change of the transducer surface. This change is picked up by the transducer and gets converted into electric signals. This then undergo amplification, interpretation, and finally display of these electric units accounting to the amount of analyte present in the sample.
Microbial sensors are widely used in many industries. Firstly, it is used in the food industry to monitor materials in the fermentation process for carbohydrates, alcohols, and acids. Subsequently, it is used to detect traces of organophosphates and carbamates, which are forms of harmful and poisonous pesticides that can be found on farm produce. It also can detect pathogenic organisms in meat, eggs, and fish. Next, it is often used in the environmental sector to detect non-biodegradable heavy metals in wastewater. The accumulation of these heavy metals may cause various diseases among living organisms. The sensor uses genetically engineered bacteria to produce measurable signs when it comes in contact with these living things in the ecosystem. Lastly, microbial sensors are commonly used in clinical diagnosis because it is a rapid, accurate, and an inexpensive way to detect pathogens, DNA, and hormones. It also can be used to track post-surgery progress to dissolve pressure sensors from the brain. These sensors can control implant infection and inflammation. The use of microbial sensors in the medical field minimizes and removes the need for physical checkups.
There are a bunch of advantages of using this biosensor. In the meanwhile, there are disadvantages too. Let's start with the advantages. Firstly, microbiosensors are cheap because they don't need expensive enzymes. Secondly, they have high stability, which means they can work reliably for a long time without losing their ability to detect substances. Lastly, the receptor in microbiosensor have the capability to be restored. This allows the microbiosensor to maintain their sensitivity and functionality over time, even after prolonged use. For the disadvantages, they can be slower at giving results compared to other type of sensors. They also have low selectivity, which means they lack the ability to selectively detect specific analytes with high precision. Lastly, the presence of cell wall in microbial receptor acts as a diffusion barrier, making it harder for the sensor to work efficiently.
Is there any latest issue related to microbial sensor?
Yeah, there are some key issues we are currently addressing. One of the issues is improving their sensitivity and specificity, especially in complex environment. We as researchers are developing new designs with advanced materials to help with detect more accurate results.
Could you further elaborate the points?
These sensors can adapt to different environmental conditions like change in pH or temperature. We strive to create more resilient designs to ensure reliable performance. Lastly, interpreting the complex data from these sensors can be hard, but we also aim to research AI-based tools for better data analysis and standardization, so that it can help microsensors become more effective.
Thank you, Dr. Jia Xuan.
Microbial sensors have achieved an advancement of being used in the biosensor chip technology. This is also known as the lab on a chip. This device can integrate numerous lab functions onto one microscale platform because it is portable and more convenient. It is used in the medical diagnostics to diagnose complex blood DNA mutations before a disease can show signs of early development. This device can also be used to detect analytes at a ultra-low concentration through active transport of target analytes to the surface of the microbial sensor. Besides that, microbial sensors have made advancements in forensic identification. These sensors will be able to detect if an agent has been genetically modified or chemically treated to make it more virulent or mask its characteristics. It is also able to detect different components like fingerprints and blood samples that can be taken from crime scenes. However, research is still being carried out to address the limitations of microbial sensors in forensic identification.
Thank you, Nick, Dr. Lee, Dr. Lauren, and Dr. Jia Xuan for sharing your insight on microbial sensors. These sensors have revolutionized on how we monitor substance in food safety, environmental protection, and clinical diagnostic, highlighting their importance in tackling our modern challenge. Looking ahead, we believe that advancement like lab on the chips and forensic application will make microbial sensors even more essential in scientific innovation. Understanding this development helps us appreciate the technology that enhance our safety and efficiency. Stay tuned for more updates on biomedical technology. This has been Shannen from Taylor University from the latest news channel. Thank you for watching and see you next time.